Hello, my name is Guillermo Parda. Today I would like to talk to you about immunohistochemistry. Immunohistochemistry is a technique that involves the use of antibodies. Antibodies were discovered in the late 1800s by Dr. Emil von Behring. Dr. Emil von Behring was working at the Pharmacological Institute of the University of Bonn, where he assisted Robert Koch, which pioneered studies in bacteriology and was a future Nobel laureate. During this time, he did various studies in which he injected animals with diphtheria bacteria. The animals would then produce what he would call antitoxins. These antitoxins would later be renamed as antibodies, for it was the way that the animal defended itself against the bacterial infection. It wasn't later, though, until the 1940s, where Dr. Albert H. Kuhns discovered how to use the antibodies to identify and mark structures inside of the tissues and cells. Immunohistochemistry, or IHC, is a technique that utilizes the specificity of antibodies to attach to a tissue or proteins, enabling the detection of a protein or tissue of interest in the sample. In the image here, we can see a negative or control state in which the antibodies do not attach to any of the structures of the cell. In the nuclear label, we can see that the antibodies have a specificity that attaches to structures inside of the nucleus of the cell. Cytoplasmic refers to that the antibodies will stick or attach to parts inside of the cytoplasm. Membranous means that the antibodies will stick or attach to parts inside of the cellular membrane. The way that this protocol functions can work one of two ways. The first way involves a primary antibody that has a fluorescent dye or a hapten. The primary antibody specificity will attach to the desired tissue or protein inside of the cell. The fluorescent dye will then illuminate itself once placed under the microscope. The second way of doing this involves a primary antibody with no fluorescent dye or hapten. Instead, it is a secondary antibody that attaches to the primary antibody that has a fluorescent dye or hapten. This method allows for further specificity of the tissue that we're targeting. In order to perform immunohistochemistry, or IHC, a sample, tissue, and staining kit are usually required. Kits usually include the same components all around. For the immunohistochemistry protocol, the first step, if necessary, is to deparaffinize, rehydrate, and quench endogenous enzyme activity. These will all stabilize and prepare our tissue for the rest of the protocol. We then must wash the slides with distilled water 3 times for 2 minutes each time. This will get rid of all the chemicals we used in the previous step. Next, we must incubate sections with proteolytic enzyme at 37 degrees. The incubation time should be determined for each application. This step will ensure that we're ridding ourselves from any enzyme activity inside of the tissue. Afterwards, we must rinse our slides with PBS 3 times for 2 minutes each. PBS stands for phosphate buffer solution. This solution can not only be used to wash or rinse, but also to dilute our primary antibody and our secondary antibody if needed. After this, we can prepare our slides and controls and perform a peroxide blocking step if necessary. Next, we incubate the section for 10 minutes with the serum blocking solution. The serum blocking solution includes nonspecific antibodies that can attach to all parts and components of our tissue or cell. These will allow for a more direct path for our primary antibodies to attach to the desired parts or sections of our tissue or cell. After this is applied, we may drain and blot it away. Next, we can incubate the primary antibodies on our tissue for 30 to 60 minutes at room temperature. After this, we wash away using the PBS. Next, we incubate for 10 minutes using the secondary antibody and wash again with PBS. Afterwards, we incubate for 10 minutes using the streptidin enzyme conjugate. This step will allow us to create a bridge between the secondary antibody and the fluorescent dye. Afterwards, we can wash with PBS. Next, we can incubate for 5 to 10 minutes using the substrate chromogen mixture. This will act as the fluorescent dye that will shine under the microscope after we're done with our protocol. We can now wash this with PBS solution. Finally, we can apply the counter stain with hematoxylin and mount the slide using an aqueous mounting solution. One of the applications for the IHC protocol would be from this study from the Journal of Pathology and Transitional Medicine. Here they observed the HER2 protein, which is present in 20% of invasive breast cancers and acts as a predictive marker for HER2 targeted therapy. HER2 testing is done via IHC and in situ hybridization for equivocal cases. Here are the results from the study. Although it may be a long paragraph, the paper suggests that the use of IHC is able and recommended to deliver more accurate results for the HER2 status determination. Here are the results in images. In picture A, we see a negative control. In picture B, we see a HER2 IHC negative. In picture C, we see HER2 IHC equivocal, which is when overexposure happens when doing the protocol incorrectly. In picture D, we see the HER2 IHC positive. These were the results that the study wanted to show. As we can see, the HER2 protein is going to be present around the cell, which is going to be in the cellular membrane. Here are the references used to make this presentation. Thank you.